Whales, despite being aquatic, evolved from land-dwelling ancestors. Fossil evidence and DNA analysis show that modern whales share a common ancestor with hippos. A phylogenetic tree helps illustrate this transition, showing how early land mammals gradually adapted to marine life. This example demonstrates how scientists use phylogenetic trees to trace evolutionary history.
Phylogenetic tree is a branching diagram that represents the evolutionary relationships among species. Each branch corresponds a lineage and merge to indicate common ancestors. These trees help scientists visualize how organisms are related through evolutionary history.
Phylogenetic trees are crucial in evolutionary biology. They help scientists understand species evolution, predict traits of unknown organisms, track disease outbreaks like COVID-19, and support conservation efforts by identifying species at risk.
A phylogenetic tree has three main components: branches, nodes, and leaves. Branches represent evolutionary pathways, nodes indicate common ancestors, leaves represent existing species. Trees can be rooted or unrooted.
Rooted and unrooted trees.
Rooted trees shows a single ancestral lineage at the base, helping trace evolutionary time. Unrooted trees illustrate the relationship without consuming a common ancestor, useful for comparisons without implying evolutionary directions.
Different tree types highlights different aspects. Cladograms shows the relationships between considering branch length. Phylograms represent evolutionary change through branch lengths. Denograms emphasize clustering, often used in classification.
Scientists construct trees using morphological data such as physical trace and molecular data like DNA, RNA, and protein. Computational algorithms analyze these datasets to determine evolutionary connections. Species with more genetic similarities appear closer together in the tree.
DNA sequencing has revolutionized phylogenetics. By comparing genetic codes, scientists can accurately determine an evolutionary relationship. Even small DNA difference helps estimate when species diverge, making molecular data essential for reconstructing evolutionary history.
In many phylogenetic trees, branch lengths represent evolutionary distance, the longer branches and shorter branches. Longer branches indicate more genetic change over time. Shorter branches suggest closer evolutionary relationships. This helps estimate divergence times between species.
Parsimony principles. The parsimony principle states that the simplest evolutionary explanation, requiring the fewest changes, is often most likely. Scientists use these principles to build the most accurate phylogenetic trees by minimizing unnecessary assumptions.
Homology and analogy. Homologous traits come from a common ancestor, examples limb structures in humans, whales, and bats. Analogous traits result from convergent evolution, where unrelated species develop similar traits. Example, bird wings and bat wings. Distinguishes these traits prevents misinterpretation of evolutionary relationships.
Convergent evolution. A well-known example of convergent evolution is the independent development of wings in birds and bats. Despite their similar function, their structures differ, proving they evolved separately rather than from a common ancestor.
Case study human evolution. Phylogenetic trees trace human ancestries, confirming our evolutionary connections to great apes like chimpanzees. Fossils and DNA studies have identified key evolutionary milestones such as bipedalism and brain's expansion, shaping our understanding of human origins.
Unlike traditional inheritance, horizontal gene transfer or HGT allows genes to move between species, particularly in bacteria. This process complicates phylogenetic trees since genes don't always follow a strict lineage. HGT plays a major role in antibiotic resistance and microbial evolution.
The Tree of Life project is a global scientific effort to classify all living organisms into a unified phylogenetic tree. It integrates data from bacteria, archaea, and eukaryotes, mapping the evolutionary history of all life on Earth.
Phylogenetic studies face challenges such as hybridization, like species mixing, common in plants and animals. Gene loss, some species lost ancestral genes. Incomplete data, requiring scientists to refrain data for more accurate evolutionary trees.
Practical application of phylogenetic trees. Phylogenetic trees help track viral evolution, such as COVID-19. By analyzing virus mutation, scientists monitor spread, adaptation, and evolution, aiding vaccine development and public health strategies.
Digital tools for phylogenetics. Scientists use software like Mega, Beast, and Phylosu to analyze genetic data and construct phylogenetic trees. These tools apply computational models to visualize evolutionary relationship accurately.
The relationships are often represented in phylogenetic trees, which illustrates pattern of descent and divergence over time. Scientists use phylogenetic to understand biodiversity, trace the evolution of traits, and classify organisms based on shared ancestry.
Phylogenetic trees are powerful tools that help us understand life's evolutionary history. As research advances, these trees will continue shaping our knowledge of species origins, diversity, and evolutionary patterns.